Okay, I want to thank the organizer for the opportunity, especially Laura and Candy and Hong, who helped me in this process. The product actually I'm presenting, how do you move this? Okay, so we are a team of investigators in the field of metabolism, as well as genetics and drug development. The product I'm presenting today is actually a therapeutic for diabetes that addresses a root problem. And that is that the current therapies actually do not stop underlying destruction of the pancreatic islets. You cannot hear? Okay. So diabetes is well known to you guys. I mean, 460 million people are actually globally are affected by it. And the GLP-1 receptor agonists that are very popular these days actually do not stop underlying destruction of the islets. There is no approved therapy actually that halts or reverses the diabetes. If you look at that slide here, you can see actually liraglutide could not actually stop destruction of the beta cell islets, actually is shown in green. So the solution that we provide here is a modified, long-lasting, catalytically inactive analog of human pancreatic chymotrypsin like elastase or CELA2A. It's actually genetically validated. Loss of function mutation in the gene that encodes this protein causes diabetes in multiple families and metabolic syndrome, including coronary artery disease. The CELA2A itself, the native protein, is a digestive enzyme that we found actually circulating in the blood. It enhances glucose-dependent insulin secretion, directly protects pancreatic islets, enhances sensitivity to insulin, and reduces inflammation. So it is actually a natural protein that rises in the blood after food intake, so shown here, and therefore is actually a safe protein. It actually secreted in complete association with the insulin levels. And in fact, actually in rat islets, it has been shown that increases insulin secretion, shown in the red line here, only when the glucose is elevated. When we knock out this gene in mouse have a smaller islets and they have higher glucose and low insulin. And we can actually rescue these mice by giving the drug to the mice or actually transplant them with the macrophages that contain this protein, etc. So this patented product protects the islet structure and number better than native protein. So it's modified protein, is better than the native protein, improves insulin sensitivity and requirement for insulin, suppresses pancreatic and systematic inflammation, and shows promising effect on reducing body weight and plasma lipids. Shown here on the right are the human islets. On top are the diabetic islets that are full of inflammation. Those are inflammatory markers in the two middle columns. The wild-type protein reduces that, but they never work as good as our analogs, which are two shown here, which reduces complete inflammation in the islets. We actually have a receptor for this protein. It's a very famous receptor, PAR2, which has been very popular with the problem that PAR2, although increases insulin secretion and survival of the pancreatic islets, it causes inflammation. Our product is the only biased agonist that reduces inflammation as well. So our product is resistant to plasma proteases, therefore safe, because they are enzymatically inactive, as shown in the curve here, with the effect of trypsin that did not increase the elastase activity. And they have a longer half-life time, as shown here. So compared to the GLP-1 receptor agonist shown on the right table, our product actually has islet protection effects. It reduces inflammation. It can be used with GLP-1 receptor agonists at a time that a patient requires insulin and is potentially curative. The foundational intellectual property, the patent, has been filed. So we would propose that actually we use this Blavatnik Award for conduct efficacy in another model of the diabetes, starting with mice, and then look at the toxicity of the drug. Obviously, we have to complement this with the available funding that we have from NIH and future funding from the industry. And our path is toward translation, to secure fund for initial human trials and pharma partnership. So if I summarize, CELA2A analogs are first-in-class therapy designed to preserve beta cell function and halt disease progression, have the genetic proof and understand mechanism, patent and safe analogs, and a clear clinical developmental path. We're seeking a strategic partnership to scale up manufacturing and advance this promising therapeutic into clinical trials. Thank you very much for your attention.
Great. Thank you very much. All right, so with that, we have 3 minutes for questions. Judges?
Does this just preserve beta cells, or does it increase beta cell mass? And then I have a follow-up question.
Both. Numbers, mass.
Okay. And in terms of standard of care, you've got metformin, SGLT2s, GLP-1s. How are you going to actually show benefit of improved beta cell mass
Okay. I showed
in
you
clinic?
Obviously, the time was short, but I showed you a slide that GLP-1 receptor agonists work as long as you have islets. Your destiny as a diabetic is that you're going to lose your islets.
Yeah.
And then one day that drug that is so popular is not going to work. So that's how you actually preserve these islets. It's actually GWAS has shown association with diabetes. In the family, anybody with that mutation in the larger family has 160-fold increased risk of diabetes. There are association with actually pancreatitis that can cause diabetes. This is solidly actually based on human genetics.
Okay, so I guess so glycemic control is steady with GLP-1s over a number of years, and then it falls off because you're not preserving islets.
Absolutely.
So are you thinking that clinically you develop this in post GLP-1, no longer glycemic controlled patients, or how are you thinking about that?
What I'm thinking about, I mean, if I heard your question correctly, is that this drug is going to be initiated once a patient requires insulin, which means that you're having defect in the beta cells. That happens to people who actually initially insulin resistant, then became diabetic. At that moment, we start, so to preserve the function, you can use that with GLP-1 receptor agonists because of the multiple benefits that they have, such as lowering blood pressure, et cetera, et cetera. So it could be a combination treatment. But at least it's going to make you to continue using that drug because then you have islets to actually cause insulin secretion.
You may have mentioned this, but what percentage of diabetic patients have the mutation?
So here's the thing. The GWAS that has shown is obviously the common variant. So this is a large population. But those are small effects that you can know from GWAS. But GWAS is a kind of way to understand the disease pathogenesis in the population. The families that we discovered are rare. I mean, we are being contacted in my lab by people who actually diagnose them and then send their blood and say, we have people like this and this phenotype, etc. But these are rare variants, obviously, probably a fraction of %. But that's not only for them only.